For the organizer to actually invite me to give a talk here. Actually, this is my first ISMB meeting, and it's really great because biologists get a chance to talk to computational biologists. So basically, I had more chance to actually think about possible lots of suggestions and advice towards us to how to improve the data analysis part. So basically, today I'm going to talk about so far, during the past couple of years, we developed this method called genome-wide in vivo RNA structure profiling, and actually a couple of labs around the world also doing similar kind of genome-wide profiling. But how actually towards the data tell the RNA secondary structure is actually quite a long way. So I'm going to actually share some actually progress in my lab and how we think about this program. So basically, before our method development, there was been pretty much 20 years people try to probe RNA secondary structure in vitro. So the difficulty to do that is actually you needed to synthesize a large amount of RNAs in the test tube, and you have to mimic a folding into a certain condition, buffer condition, so it will fold very stable, so you will be able to do the enzyme or actually chemical modification on these RNAs to generate RNA secondary structure. But for a very long time, the living cell condition, the RNA folding structure is very different because you associate with lots of RNA binding proteins, different metal ion, also osmolytes, all these things. So it's very complicated. Plus, every single RNA towards abundance-wise, it's impossible to do the older methodology. So at that time, when I was working in a chemistry and biology lab, I was thinking, okay, we should have a better way to probe in vivo RNA structure and actually probe many RNAs in one single experiment. That would be super great. So the first approach the approach developed is from chemical probing methodology. So we're using one of the chemical called dimersa sulfate. So what it do is when your A and C nucleotide is single-stranded, so it will methylation the N1A and N3C position. So it will put a methyl group on this position. So this position is on the Watson-Crick phase. So what it means if you have a methylation on this nucleotide, it will block your reverse transcriptase. So it couldn't actually process because your Watson-Crick phase was blocked. So if you have more reverse transcription stalling at this position, that means you will have a high probability this nucleotide will be single-stranded because it exposed to the dimersa sulfate a lot. So we generate a term called DMS reactivity. So if you have a high DMS reactivity, that means lots of reverse transcription stalling, then you have a high probability to be single-stranded. Opposite, if you have a less stalling of reverse transcription, of course, it will have a high probability to be double-stranded. So along with the dimersa sulfate, we have another chemical, actually synthesis from Eric Kuhl's lab and also Howard Chang's lab in Stanford. So it's called SHAPE. So what it's doing different chemistry. So dimersa sulfate is working on the nucleotide base, but the SHAPE NAR is working on the acylation of the 2' hydroxyl group, so make a big block on your RNA. So it also can be taking the signal by the reverse transcriptase, so it will block. So the difference between two chemicals is SHAPE can target AUCG4 nucleotide. So if it's similar to the DMS reactivity, so if you have a high SHAPE reactivity, that means you have a high probability to be single-stranded. Opposite, you have actually high probability to be double-stranded. So originally, we developed the whole method using a model plant called Rhabdotocediana. So we developed the method along with also Jonathan Weissman's lab in the East. So we published a paper together actually on the DMS probing structure. So now all these two chemicals can apply in my lab. So far, we applied in East, C. elegans, human, also different type of a plant, wheat, rice, these kind of plant. So all these chemicals can be penetrated in vivo into all these different species. So how the deep sequencing pipeline library construction here going through, this is the detailed pipeline. So you basically treat your cell or plant or even the yeast or HeLa cell with the chemical for a while, then till the chemical can penetrate it into the cell and modify the nucleotide. So you generate, you can do different selection of poly selection, ribosero selection, or these different things, selection to enrich your RNA of interest, then you do the reverse transcription. We use the N6 random nucleotide to tag Illumina primer sequence to be able to do reverse transcription. So we generate a single-stranded DNA. So we put a single-stranded DNA adapter in the 5' end. So we generate a double-stranded library of a deep sequencing library. So because everyone knows possibly there's lots of background towards reverse transcription random store. So that means for in the no treatment library, you're still gonna actually have lots of signal from the reverse transcription stalling in the middle of your gene. So also we prepare the parallel, another minus chemical library. So to using that to subtract the noise from the plus library. So basically, this method is really powerful. Previously, this is about a 3,500 nucleotide 25S ribosomal RNA. This is a phylogeny derivative structure. Previously, we needed to run pretty much 20 joules experiment to actually get a really deeper, the whole RNA structure information, and it's not a nucleotide resolution. But with this method, one single experiment, you can get a really good nucleotide resolution of the structure information. So this is, and the reactivity agree with the phylogeny structure, which is supposed to be the closest structure to the in vivo very well, because you can see the single-strandedness, you get a high DMS reactivity, but the double-stranded area, you have a low DMS reactivity. So also there's another powerful part with this method is because one single experiment, you can get lots of RNA structure information. So this is a rhabdosis we achieve pretty much 24,000 RNA structure information with nucleotide resolution. That means every single nucleotide will have a structure information and a reads count. And you have 10,000 which have a 10 reads per nucleotide and nucleotide resolution of RNA structure. So this is the web browser we give it to the whole community to actually to search their genes, and further on we will actually put more species for RNA structure library into the web browser. So basically, because of this powerful tool, so people then starting ask these questions people really want to know for like 20 or 30 years. These actually how the role of RNA structure in these kind of RNA biology process. So here I just put several examples of how we actually think there will be actually strong role of RNA secondary structure in these RNA biology process. So first we go for is a translation. So where you have a 10,000 transcript RNA information, the immediate thing you want to do is try to do some metagene analysis. So what we did is we align start code and also align the stop code and average all the DMS reactivity crossing 10,000 transcript. What you can see, there will be actually high reactivity upstream start code, which is representative for more single-stranded region. So we think this is correlated to the ribosomal initiation. And also very interesting part is in the coding region, you're starting having these kind of 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3 pattern. And which is very puzzle. It's only in the coding region. If you actually summarize all these position of a codon unit, you will see the first position of your codon have a high DMS reactivity, more single-strandedness. The second part, second position is actually more double-stranded. Third part was more media status. And this also showing in the shape data, which I didn't show here. So basically, when we do the Fourier transformation, you can see it's actually very obverse only in the CDS region, not in the 5-UTR or 3-UTR region. And this pattern is really associated with high translation. So in the low translation group, you don't see this kind of pattern. And another interesting thing is that people will say whether or not your signature of this periodicity is due to the ribosome periodicity, so ribosome profiling data, whether or not it's ribosome protection there, but actually we look into the ribosome profiling data, it's actually out of our expectation. So the first position, which have a more single-strandedness, actually have a more association with ribosome. So that means for all these structure is actually ribosome naked structure, ribosome-free RNA secondary structure. So that means pretty much maybe structure actually determine these ribosome periodicity as well. So then, of course, we go for some literature, try to find the alternative polydylation site, which lots of people propose it will have a structural. So what we did, so previous study on the alternative polydylation, all the alternative polydylation site will have an AU rich region just upstream of a cleavage site. So when we compare our structome, all these regions tend to have a strong structure with the AU base pairing. So we think there will be actually possible a strong structure signature just for the recognition of alternative polydylation. Also, lots of people in the audience interesting is the alternative splicing. So because this is the first library we generated in the mature mRNA, so basically we only can capture exon information. So what we did is we took the 5' splice site, 100 nucleotide at the junction of exon intron region, and we separate the splicing events to the splicing, successful splicing events, and also unspliced events, which including the intron retention and exon skipping. So what you can see is that the unspliced event tend to have a very strong structure in the minus 35 to 40 nucleotide, and we have a very strong structure block here. And if we did a nucleotide chiffering, we didn't actually see this kind of pattern. So a nucleotide composition control, we didn't see that. So that means this proportion is actually due to the structure itself. But now in my lab, we actually pull down the nucleus pre-mRNA structure, try to understand whether or not this structure is formed itself in the exon or it's inherited from the pre-mRNA. So that will be something later on, hopefully we can talk later. So here, because this is ISMB meeting, I really want to actually show some details towards how we understand these kind of new deep sequencing data and how actually it's possible to give a more accuracy prediction of RNA secondary structure. So how to, from these kind of experimental data towards reactivity profile, and also later on the final things which we really want to see as a secondary structure. So at the very beginning, when you gather your deep sequencing data, you want to compare a biological replicator. Lots of people doing RNA-seq, they will check RNA abundance. So this is one step we always checking whether or not the RNA abundance is quite similar in both biological replicator. But it's not enough, because you really want to know whether or not your chemical modification library is really going to be replicated the same modification as in different biological replicators. So we normally take all the ribosomal RNAs information from the library and try to compare the minus group and the minus group with minus group biological replicator and also plus group. So what actually happening is because the chemical modification counting file on your ribosomal RNA is very specific. So that means your biological replicator should have a very specific, very high correlation because it's very specific modification of your nucleotide. But the minus group is more kind of a reverse transcription random store, so it should not actually have a high correlation. But also, between the minus and the plus group on the ribosomal RNA counting file, you actually will see a very bad correlation because one is actually chemical specific modification, one is actually your reverse transcription background. So based on that, we actually carry on with how to actually do something with the deep sequencing raw data. So here is a traditional method of how we measure the gel assay. So this is a pretty much older standard way how you actually quantify an RNA structure. This is a sequencing line of AUCG. Pretty much this is a minus chemical plus chemical. What you really measure is how the band intensity compare in the plus lane and to the minus lane. So as a traditional way to measure the band intensity is you actually measure each band intensity and normalize to the whole length, all the bands crossing this lane, and get a normalized intensity. And then using the plus normalized band to subtract the corresponding minus normalized band, so which you generate a chemical modification rate. So this way is actually how important the two things in this kind of generation, the chemical modification rate is you basically can see the full length here, which is very essential for notice how to normalize each band's normalization, because this full length gives us a very bigger, it will become a bigger denominator, in a very bigger part of your denominator. So this is one thing you can notice, the full length band is very important. Second band is all about how much more specific on the plus lane towards minus lane. So the normalization is very essential to know whether or not it's a true signal or actually noise. So then based on that, when you transform to the deep sequencing data, you will notice on every single transcript, the reads counterfile comes out very skewed. So some are thousands reads in one position, and several positions is maybe only 1 or 2 reads. Then the trouble is that how data is very skewed, we have to do some transformation onto these reads counterfile. So of course, we did a log formula for these kind of data. So what we do is we take every position of the plus reads, take the log formula, then normalize all the reads, including the full length in the whole transcript, and subtracted the minus group. So this is the way we actually derived this equation from the traditional gel assay. So we generate a reactivity profile like this kind of log formula. So there's another parallel formula which lots of people really like. Also previously we really like as well, is actually a max likelihood formula, which is counting the probability of a modification. So what it actually do is every single position have a plus count, so how many stalling counterfile, and divided how many reads were actually passing through this stalling position, and you actually did a subtraction ratio of the probability. We previously really like it because it's really neat, it's really counting the probability. But however, Illumina sequencing had a really limitation because the longest fragment is about 600, 650. So even you do the peri-end mapping, this denominator is only the fragment of total reads count. So you don't have the ability to do the full length normalization. Plus, this way to do that is the data itself very skewed. So when we plot a DMS reactivity, or this is shape reactivity, you see crossing MLE, max likelihood formula, you see pretty much very skewed reactivity profile. And the y-axis go crazy. So the trouble with this kind of method is when you do the metagene analysis, you don't really know what's the real signal because this kind of reads distribution. So also, if you plot the distribution of the reactivity crossing the whole transcript, you see log formula have a quite nice distribution, but the MLE is quite skewed. So MLE also applied for these actually reactivity calculation, which don't using the log formula. It will also leading to these kind of a very skewed distribution. The trouble for these kind of normalization is you increase the true positive. So that means if you really single-stranded, the accuracy of single-stranded is high. But however, you increase a large number of false negative. So which actually causing a larger proportion, lots of nucleotide, suppose not a single-stranded, then become double-stranded because of this kind of case. So also there's another thing towards whether or not we should have a full length reads there. Also we should do a full length normalization. So basically previously lots of methods say that we don't need a full length because there's not already modification information there. We just capture all these modified nucleotide. But the trouble is then you do the fragmentation, so you basically just know whether or not this nucleotide is modified. But however, RNA structure is more like the relationship between each nucleotide crossing the whole transcript. So you really want to have a very detailed understanding about the relationship from the first nucleotide towards the end of nucleotide. So if we do a full length normalization, what actually comes out is you can see these kind of a structure island, you have low reactivity, then you have actually a very high DMS shape reactivity, then you have a lower, then you have a higher. So you have this kind of relationship across in your whole RNA. But however, if you do the fragmentation of your library preparation, also you only do the reads around the region to do local normalization. The trouble for that is all these low reactivity bring up because of the normalization method. So crossing the whole transcript, you didn't see previously this kind of a pattern. And surprisingly, if you just fold it, it gives you complete different structure. So the trouble is that how to accurate to know how these relationship, the full length is really important normalization to actually really know what's going on of your RNA. So there's another thing which hidden information towards normalization steps in every single structure paper now is, lots of people, especially the computational biologists, don't know why. Because lots of people doing 2-8 normalization at the step after you generate the chemical reactivity. The reason why why we're doing 2-8 normalization is because previously when we run these kind of gel assay, because the gel assay were stacking very strongly, so squeezing a lot at the very beginning of your gel. These nucleotides is really hard to separate. So the intensity here is super high. So that means this cannot be really accurate to measure it. So that's why we do, after generate reactivity, gel intensity, you will do a 2-8 normalization. So you sort of top 10% high intensity of the band. Then you take the top 2% away. So these kind of outlier away. So then you're using the bottom, the rest 8% as a denominator to normalize every single band of your gel intensity reactivity. So this way is actually derived from the experimental gel assay. But when we actually apply for the deep sequencing, you can do 2-8 normalization. You also can do 5 to 10% normalization. Also, you can pretty much do in box plot. It will give you quite a similar actually structure information. So from that on, then the next thing is you want to plug your DMS chemical DMS or shape chemical reactivity into the RNA structure or the VNF fold. Try to fold your RNA secondary structure to know whether or not actually the in vivo looks like. So there's quite a standard now is using Kevin Weeks and also Dave Matthews' lab in North Carolina. They actually did a lot of things towards this term called pseudoenergy. So why it's pseudoenergy? Because it's not the real energy. So it was actually becomes an additional penalty score for the normal free energy. So let me just walk you through how we actually plug a shape reactivity into it. So basically, you will actually, the pseudo-energy will generate by this equation. So you have M and B parameter. Then every single shape reactivity of every single position will plug into it, then take a log formula, generate a pseudo-energy. So this is the HIV part of RNA. So the pseudo-free energy only constrain the base pairing site. So this is a helix plus unfavorable loop. So when you calculate your delta free energy, you actually calculate both the stacking energy plus this kind of entropy penalty score for the unfavorable loop sequence. So you generate a delta G. But when you plug the shape pseudo energy into it, you actually only constrain these base pairing region. And on the end of the helix and the middle of the helix, it will have different actually power of actually constraint value. So then you generate a total delta G, which have your thermodynamic delta G plus pseudo energy. But however, because this is a very simple equation, so all your reactivity crossing the whole transcript need to follow a good reactivity distribution through your RNA structure. So basically, you have to actually have each RNA have a really good distribution which are crossing the whole RNA. So this requires a good transformation. Like I mentioned before, the log formula will be much more better for these folding pseudo energy. And also, there's another thing is that how you actually determine the M and B. So every single structure library will need a certain standard actually internal control to know is very detailed whether or not this MB is really true for your library. So what we do is we're using the standard RNA for 25S ribosome RNA, also 18S ribosome. This is a plant one. Also, every different organism will have their 25S and 18S ribosome RNA. Using this phylogeny structure as a standard to fold your structure with your constraint and with different MB to actually counting how much similarity between your constrained structure with the phylogeny structure. And also for the studying the RNAs in the nucleus, because you don't have a ribosome RNA, so what are you using is U12S RNA as our experience. So this works very perfect well. So phylogeny derivative Arabidopsis, this is U12S RNA. So the shape and the DMS reactivity agree with it very well. And in vivo, you see a protein protection site here. So we can actually really analyze how the folding really actually control to training the folding to agree with these phylogeny structure. So basically, using this equation, we're training the M and B value. So generate a kind of a score, the highest score, which means the similarity between your phylogeny structure and your constrained structure. If you reach the highest one, this is the one you're choosing to fold all the rest of your RNA. So far, this is the way how to accurately to actually determine the structure in your library. So based on that, of course, when we have 10,000 mRNA, we can have a constrained folder of RNA secondary structure, also the in silico folding structure. So we did a quick comparison towards how much difference between the in vivo structure and the in silico structure. If you see, this is about a 10,000 transcript. If we just compare how similarity between the in vivo and the in silico, majority actually RNA is tended to fold differently from the in silico prediction. There's a high PPV, which means high similarity. So in silico and in vivo is quite agree. Low PPV means actually quite different. And to our interest is actually this kind of difference had a reason, because all these very different towards the in silico thermodynamic stable structure is tended to actually have a stress response, also temperature, especially the temperature response function. And all these very stable structure, which is similar to the in silico structure, had a quite a fundamental translation cell maintenance function. So this is actually give us a clue is if in vivo structure is very different to in silico prediction, and may actually have a regulatory rule. So take-home message from my talk is, first, we have these powerful methods which we can do the in vivo RNA structure profiling. And the very important thing is that it's not a simple application, simple method to use. You have to actually think very accurate towards how you do your control, how to actually using these kind of, whether or not using log of transformation for meta-gene analysis, or actually using MLE or these local formula, analysis for these individual RNA or fragment of your RNA structure. And also, it's very important to have a full-length normalization, especially the full-length band. It's not a sacrifice. It's actually really important for determining the correct structure. And plus, you have to have the correct training of your pseudoenergy to make sure that it gives you the right constraint to put into your delta-free energy. So this is my group. I just get a chance because my group have 4 bioinformatician, 4 white bench person. And we still don't think it's enough to have enough support from the bioinformatician in the lab. So it becomes more dynamics, and actually it's really good because the bioinformatician, also computational biologist, also modeling people can have lots of good suggestions towards white bench people to actually design different experiments to actually understand the question. So if anyone or just graduates actually interested in my lab can come to talk to me. Thank you.
Time for questions.
Great talk, thanks very much. I'm just speculating on the 3-nucleotide pattern in your ribosome profiling. Could it be that since you're modifying A and C by methylation, that it first of all blocks ribosome, and then the first and the third nucleotide are simply more exposed to further DMAS modification, and the internal one is simply buried in the ribosome, and this is why you see this V-shaped pattern?
So actually, we had the shape data, which actually is quite primary data. So what we saw is we also, in the shape data, we also see AUCG, so as periodicity. So that is not due to the AC composition. But the interesting part is that when we really compare detail with ribosome profiling data, so the interesting part, the periodicity is not actually strongly at the very beginning. So it's getting stronger till the end of your RNA, which actually you can think about the ribosome actually moving faster crossing your RNA. And the periodicity more stronger, the ribosome actually moving more faster. We actually, so it's harder to, we think it's not it's the reason because if as you see on the starting position also, just to go back a little bit. So basically what we saw is if it's ribosome protection signal, then because if you see, sorry. So if you see this position, actually around this proportion is actually high accumulation of ribosome. Ribosome is about 30 nucleotide occupancy. So that's why normally if you have a correlated to the ribosome occupancy, you won't see any single strandedness here because it will be highly protected because that subunit is super big. That's why we think a majority structure we capture here is ribosome free structure, because a structure is actually a majority structure average signature here. We can talk later.
Other questions?
It's a very nice talk. So quick questions about this. So did you try nuclear fractionation and looked at the signals?
Nuclear fracturing?
Yeah, so only taking things from the nuclear.
If we only take the nuclear composition, we don't see really strong periodicity.
Okay, so that's like
Yeah,
a
yeah.
sanity check about your, okay. And so, so you sort of describe the computational pipeline that you have. And what I was wondering, because you said there's a lot of challenges, but the way you presented it, it seemed like, okay, you have it already. So where are your, so what wasn't clear for me was where do you find your problems right now? Where are the main challenges? What are you not happy about?
So the problem here is when we define shape or DMS reactivity, it's actually a reactivity we think it's a representative of probability, right? But however, when you really fold the RNA structure, you still need to go through pseudoenergy, which means how the pseudoenergy is capture your shape or DMS reactivity. So towards the sensitivity-wise, it's not really that sensitive because you have a rank of a rate of how many actually single-strandedness is actually there. So when we actually do that is, so far we think it may be possible there will be a way to develop a method to be able to actually go from the very beginning till the end. You only have a one probability value, that's done.
So I'm wondering about your ratios, right? Because you in both cases, whether you do the likelihood-based ratios and where you do the log transformation, you're always looking at the ratios, and with the log transformation, you're squishing out the really big signals, right?
Yeah.
But what seems to me when you were describing it is that if you didn't see a lot of information going both ways, instead of, right, so your fold changes.
Yep.
The way you're doing this. The log transformation is not going to help you. If the total number of observations is very small, it's going to be very, very noisy. And your model, because it's only ratio-based,
Yep.
is not allowing for that. So it seems like you need, instead of a point estimate, you want to build a posterior distribution where you have more or less belief in terms of what is the ratio.
Yeah, and to do that, the most difficulty is so far only have a ribosomal RNA, phylogeny structure, or a little bit of small RNA, snRNA we can using as actually a standard to do that. Otherwise, you still don't know how actually ready to see that. So that's the limitation for actually doing a better model towards actually just using that way. So it's kind of limitation towards a certain amount of RNA using for the standard, but you still don't know whether or not if it's true for your ribosomal RNA, whether or not it's true for your mRNA. So that's the part we still kind of try to seek out. But however, now we have an individual library just for individual RNA. So then you can see more mutation, more temperature treat. So then maybe there will be a way to actually tell more towards that information. But so far, if we just are using ribosomal RNA, it's really hard to actually give a very accuracy structure prediction on other non-ribosomal RNA. Yeah.
So I was very interested in your last bit of information about the stress response genes and the heat shock genes that you mentioned. Are they overall more or less structured than other genes? And what happens if you do the experiment at elevated temperatures? What would you see, or what would you predict that you see?
So basically, if we saw, so we did also temperature library, but actually it's quite preliminary data, so I haven't to show here. So it actually give you different type, even in the heat shock condition, like we treat it from 22 degree to 37 degree. So 37 degree is quite high for the plant one. So when we saw that there was a certain proportion, you actually single-strandedness actually more increase, you see a denaturing. But actually opposite, they actually have sometimes lots of base pairing formation crossing even heat condition. We don't know why. We think there might be potentially a switch or certain way how to stable the stem in some way. But however, it's completely different in the in vitro condition. So, which actually in vitro is more, you can see some linearly change, especially on smaller fragment, but the in vivo is not as you expected. So this is something we try to analyze. It's also a challenge as well because you have to normalize between two different type of conditions as much as possible. So we have some clue towards certain RNA, but the genome-wide we couldn't really actually see now.
Great. Let's thank Yiliang again for a great talk.